Welcome to Qlucore Omics Explorer. In this tutorial, we will show you how to import a raw count data matrix using the wizard. Using the wizard, you can import normalized data, as well as annotations connected to your data set. You can also import raw count data, which is what this tutorial will show. The wizard will guide you and help to define which blocks of a file are data, and which are annotations. This is best illustrated with an example. This is the text file with the raw counts data that we are going to import. Samples as columns. Sample annotations for descriptions of the samples. 50 rows of variables. And 4 variable annotations for descriptions of the variables. Let's start. We open the file with the Open with Wizard option from the File menu. That opens up the wizard, and we step through the wizard using Next and follow the instructions. First question: is this a raw count matrix? Yes, press Next. Next question: select Separator. This is a tab-separated file, so this is correct, press Next. Next question: matrix orientation. The rows are variables, this is correct. We can also handle transpose files with samples as rows. Press Next. Select top left data cell, and then press Next. Select top right data cell. For a data set with many columns, it might be necessary to scroll to the right to identify the upper right cell of the data. The whole data block will then be marked blue. We have now defined which part of the file contains the measured data. Next, we select variable annotation headers, include the gene length column. If you select the wrong cell, just click on it again to undo. Press Next. Select sample annotation headers. Note that you are not forced to import all rows with sample annotations. Now we are ready to import the data. The data is now imported and the Data tab is presented. To normalize your data, select from the different options. If you plan to work with DSeq2-based statistics, select VSD normalization. Here we see our 12 samples. Here we have the sample annotations that we can use to color our samples. Remember to save the data. A good choice is to use the G-data format. This will make it faster to open the data set the next time. Find more video tutorials on our website. Thank you for watching and good luck with your analysis.